Welcome to our lecture presentation on cell structure and function. We'll begin our discussion talking first about the basics of cell structure, in that the cell is the basic unit of structure and function. And what this essentially means is that when you say the cell is the basic unit of structure, it means that if you take any organ within the body, say it's the pancreas, the brain, or the heart, the structure of those organs are made of cells. So it's the cells that are the building blocks of every other tissue, organ system, organism. It's the smallest unit of life. And the function, basic unit of function, is also the cell, because if we look at, say, for example, the heart, the job of the heart is to contract and move blood throughout the body. So therefore, it's the cells of the heart that actually contract and shorten to cause the whole heart tissue and organ to contract and move blood. So when we say the cell is the basic unit of structure and function, it's the cells that we're made up of and the function of those cells that determines if we live or die. For example, we need to bring in carbon dioxide. It's the cells of the lungs that absorb that carbon dioxide and carry that, oxygen, that carry that oxygen from the lungs through the cell wall of the alveoli into the capillaries, which leads to our blood. So therefore, the cell is the basic unit of function and leads to all life as we know it. If we look at cells in general, they have three things in common. All cells have a plasma membrane. All cells have cytoplasm. That cytoplasm is the stuff that fills our cells and allows all the chemical reactions to occur, and it suspends the organelles. It gives the cell its shape as well. The organelles are the smaller structures within a cell that have specialized functions. And we talked about those in lab, such as the rough endoplasmic reticulum, the Golgi apparatus, the ribosomes. They too are membrane bound, and they have specific functions inside the cell that helps that cell contribute to that tissue, organ system, and organism. So if we look at the plasma membrane of a cell, we can see that it's quite complex. Looking at this picture here, we can see a couple of different molecules working together. Right here, this main structure of the plasma membrane, these are phospholipids. So this region here, as you can see, there's a circular region, then there's a thin, wispy region. Those are called phospholipid molecules. There's a tail portion in the core here, and the head portion are the round circles. These phospholipids are fatty molecules. So therefore, we know that oil and water don't mix. So if we have a substance that dissolves in water, it will be attracted to the surface because these heads attract water, but the tails are fatty. We call them nonpolar, and they repel water. So there's no way for water-soluble substances to cross freely from one side of the cell into the cell, and vice versa. As a result, we have to have special proteins embedded in this membrane to transport water-soluble substances. So that's what these larger structures are. Some of them serve as transporters, some serve as enzymes. There's a variety of functions that these membrane-bound proteins serve for the cell. But you can always identify the proteins because they're large and globular in structure. So we look, here's a protein, we can see here's another protein embedded in the phospholipid layer. Here's another protein, here's a protein here and here. All of these are proteins. So they, like I said, can serve as transporters, helping molecules move across the membrane that cannot dissolve in that fatty core, or they might serve as enzymes. Those are two common examples of proteins in the plasma membrane. Extending off the surface of the plasma membrane are carbohydrates. These carbohydrate chains here act as markers for cell communication and cell recognition. Essentially, these carbohydrate molecules are like the face of the cell. It allows one cell to recognize another cell. It also, the pattern of carbohydrates on the surface also will tell an immune cell when there's a bacterial cell present. It won't recognize that carbohydrate pattern on the surface of that cell, and as a result leads to an immune system response. So these carbohydrate chains on the surface, shown in green here, serve as markers for cell recognition. Lastly, another important component of the plasma membrane is cholesterol. You can see these yellow chain 4-ringed structures here embedded in the fatty portion of the membrane. Cholesterol too is a fatty substance, and as a result it is found in this fatty portion of the membrane here among those phospholipid tails. And this gives a little bit of stiffness to the membrane, because the membrane would be oily and not have enough stiffness to it to actually maintain a good covering for the cell and to protect the cell from the environment outside the cell. So cholesterol adds a little stiffness to that membrane. Just as we know if we have too much cholesterol in our diet, it can deposit on our arteries, inside of our arteries and veins, and cause fatty streaks and damage to the vessel walls. So cholesterol is good in a point. Our liver makes all the cholesterol we need for our plasma membranes and for making certain hormones. But the extra cholesterol we bring in from our diet is not necessary. Examples of foods that contain cholesterol would be anything that is an animal product. For example, fish, meat, any other kind of meat, eggs, dairy products. Those all contain cholesterol. So I have an animation here of the plasma membrane. I'm going to get out of this PowerPoint for just a minute and show you an animation of the plasma membrane. And I'm hoping the sound works. We'll see if it does. Hoping it does.
I do it to stay sharp. I take care of my body, but
well, that's not the correct video, so I'm having issues here, it looks like. Anyway, you can watch this video here. If you copy this link to your screen, you can watch this video of the plasma membrane. It's very good. It's only 1 minute and 26 seconds, so definitely worth your time to watch that video. So we'll proceed onward with our discussion here. Get back to full screen. Looking at movement through the plasma membrane, we'll talk about how substances move through the membrane. Four ways. One is diffusion. That's shown here. Simple diffusion is where a substance that is fat soluble can diffuse right across the membrane without any help. Some examples of fat soluble substances would be carbon dioxide, oxygen, alcohol, and fat-soluble vitamins like the vitamins A, D, E, and K. It can dissolve freely across the membrane, always going from higher concentration to lesser concentration. Osmosis is the same as simple diffusion, but it's just the diffusion of water, so we're going from higher water concentration to lesser water concentration. So when you see the two words diffusion and osmosis, both involve the movement of something from high to low. Diffusion is generally referred to substances that are dissolved in water, like a salt solution, so looking at salt, sodium ions, chloride ions, something that can diffuse across a membrane from high to low concentration. Again, osmosis is just the diffusion of water from high water concentration to low water concentration. Filtration is movement of a substance due to pressure differences. We see filtration in the kidneys, where we have blood pressure that helps push some of the substances, some of the fluid out of the kidney, the blood vessels of the kidney, into the kidney and then into the bladder to form urine. Mediated transport mechanisms are mechanisms that require energy, and we'll talk about that in a little bit. So back to simple diffusion. Key thing to remember here and to underline and highlight is its movement of solutes, which would be any dissolved substance, from an area of higher concentration to lower concentration. And the rate of diffusion, how fast this process goes, depends on how concentrated the substance is on one side of the membrane. We call that a density gradient, meaning it's more concentrated on one side than the other. And viscosity, how easily a liquid flows. Something with a high viscosity is something that does not flow very well. For example, molasses would have a high viscosity compared to, say, a cheap pancake syrup that flows very easily. Again, osmosis is the diffusion of water across a membrane. And this is important for us in our bodies because we have a constant shifting of water that occurs as a result of osmosis, going from high water concentration to less water concentration. Ideally, we don't want huge water shifts. We want to have equal amounts of water on either side of a membrane. For example, our capillaries, we don't want a lot of fluid flowing out of our capillaries into our tissues because that would cause swelling. And we don't want a lot of fluid going from our tissues into our capillaries because that would cause high blood pressure. So ideally, you want just a small, no major movement in and out of our tissues from our blood via osmosis. Facilitated diffusion is a process by which we move a substance from high to low concentration, as in this diagram here, but that substance is probably water soluble and cannot diffuse directly through the membrane. So as a result, those molecules have to have a protein transporter to move it across the substance, across the plasma membrane. So we call that facilitated diffusion because it's diffusion with a little help. But that help does not require energy. So it's just like osmosis and regular diffusion or simple diffusion, and that substances are moving from high to low, and it does not require energy from the cell. There's that animation here. You can copy and paste that link to your desktop. I am nervous to do that because my computer seems to not want to perform very well when I'm using this recording software. I'll try it here and see if it works. Yeah, it looks like it's going to cooperate after all. Okay, so we'll watch this video quickly on how facilitated diffusion works.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
So facilitated diffusion does not require energy. It's a movement of substances from high to low. Active transport is a little different. Active transport does require energy, where it's taking substances and moving it against the concentration gradient, which means it's going from lower concentration to higher concentration. So there's a difference. And here is an animation showing that.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
So we can see that this type of transport involves moving substances from low to high concentration. Here we can see there's already sodium outside the cell. Now this pump, with the help of ATP, so see how the ATP, the 3 phosphates come in? 1 phosphate is removed, and that powers this pump to take sodium against its concentration gradient and add more and more of it to the outside. Same thing with potassium. 2 come in to add to the potassium that's already in higher concentration inside the cell. And it's a continuous cycling back and forth. Notice that there's 3 sodium that's being removed from the cell and taken outside, and there's 2 potassium that are being brought into the cell with every cycle. And again, it requires ATP because that phosphate, that 3rd phosphate on the ATP molecule, is what powers this process. So the other steps here, the other types of transport, are all energy requiring processes. So endocytosis requires ATP energy. And endo, when you hear the word endo, think inside. This is where we're taking particles into the cell. So you can see how this cell here is wrapping its plasma membrane around this bacteria to bring it inside the cell. This might be a white blood cell, for example, fighting infection. So it's bringing in this into the cell by wrapping its plasma membrane around it and forming a vesicle. So phagocytosis is a type of endocytosis that involves solids, taking solids into the cell. The term phagocytosis means cell eating. Penocytosis involves bringing liquids into the cell. So this is translated, penocytosis means cell drinking. And receptor-mediated endocytosis means there might be some receptors, some special molecules on the surface of the cell that the substance has to bind to first before it be brought into the cell. Exocytosis is the opposite of endocytosis. It's the taking of substances from, say, here's the Golgi apparatus, and pinching off the plasma membrane and sending that substance outside the cell to be exported from the cell. Examples of that is the pancreatic cells export digestive enzymes out of the cells of the pancreas, and those go to the small intestine where they're used to help us digest our food. Mucus from salivary glands, same thing. The mucus leaves the cells of the salivary glands and enters the oral cavity where it does its work. And the secretion of milk by mammary glands. That milk travels down the ducts and is released when baby starts to suckle. All examples of exocytosis, where you're moving substances out of the cell in small membrane-bound vesicles. So that concludes part 1 of our discussion on the cell, and we'll pick up with our next video on parts of the cell.